So hello, everybody. It's a pleasure for me to introduce the speaker of this yellow webinar, Professor Carlo Jose Pirola. He's currently a senior investigator at the National Scientific and Technical Research Council, CONICET, from Argentina, and is serving both as the chair of the Department of Molecular Genetics and Biology of Complex Diseases, and as the director of the Institute of Medical Research at Buenos Aires. So his major interest is systems biology of metabolic syndrome components, including hypertension, obesity, type 2 diabetes, and fatty liver. Enjoy the seminar, please. I will pass the word to Prof. Pirola. We can start.
Okay. Thank you, everybody, for the invitation and the pleasure to be with you today. I have to declare that I was the director of the Institute until 2 weeks ago. Now the director is Dr. Silvia Sokoyan. So, sorry for...
No problem.
You can rest.
Yeah, I don't wish to borrow the title now. You? Okay, let us start. Are you see my screen?
No.
Oh, I have to. I know.
Share.
I have to. I guess share the screen. Do you allow me to share the screen?
Yes. Here. Lucia, where is invite? No. This is mine. I cannot share the screen for the others, but now our secretary will solve this. Don't worry. Why I cannot do that? Okay.
Okay.
Only a second.
I get it.
Okay.
Now I see my screen?
Yes. We see the screen.
That's okay?
Yes, it is.
Okay. I have no conflict to declare, and the talk will describe all of these topics, starting with the kind of difference between system biology and system bioinformatics. System biology formally is the study of the behavior of complex biological organization in terms of the molecular component. The central goal is obviously to infer the response of biological system to external or internal perturbation from the property of this component. So the complexity of the problem is showing in this slide. This is a kind of synthesis of the metabolism of all of living organisms. The current knowledge now is this kind of simplest schematic. This is a paper showing the model of the liver metabolism from 200 enzyme and transporter reflecting 4 compartments, almost 300 metabolites, with the parameters obviously taken from the literature, like affinity constant, maximal velocity, and so on. The model now is currently able to reflect circadian variation of the common metabolites in the normal hepatocytes, and can predict the behavior of the hepatocytes when receive a bolus of ethanol, for example, increasing lipid content and lactate in the cells. Even the point of view of diseases can predict the difference between adenoma, hepatocarcinoma versus normal hepatocytes. But the problem with these models are that the parameters were taken from in vitro experiment performed mostly in rodents, rats, and mice. And the model has evidently low complexity, lacks many gene products associated with fatty liver disease, an example, PNPLA3, and so on. So system biology tried to harness the power from the technological advancement in the field of omics and the advancement of next generation sequencing. This technology provides a spectrum of information from genomics, transcriptomics, proteomics, epigenomics, pharmacogenomics, metagenomics, and metabolomics in a bottom-up approach. But owing to insufficient experimental data, is fell short in constructing good models. So probably the talk today will include the utility of system bioinformatics, which is a kind of different approach because integrate in a top-down approach using bioinformatic methods, extracting and analyzing information from omics data. And then we have the opportunity to build an integrative profile of the human system using computational methodology like a machine learning, pattern recognition, and data mining, and so on. So the next topic will be applying this kind of approach, what the heterogeneity of fatty liver disease, which is the overlapping between alcoholic and non-alcoholic fatty liver disease. Many years ago, we published this approach and showed that both diseases has many shared pathways, mostly reflecting important physiological process like apoptosis, interleukin-6, NF-kappa B, and other signaling system, mostly related with cancer and other adipocytokine signaling pathway. So summarizing the evidence, we see that the shared genes between the two diseases are around 350, and these 350 genes are enriched in molecular function described here. This is acetylase activity, storage protein, catalytic activity, growth factor activity, and so on, cytokine activity. Regarding the biological pathway of these common genes between the two diseases, you see there are very important pathways regarding the biosynthesis of triglycerides, bile acid, TGF beta receptor signaling, BGF and BGF receptor signaling network, and so on. Another important pathway is hypoxic induced factor 1. What about the specific pathway in these two diseases? Well, regarding the alcoholic fatty liver disease, there are 3 important pathways, one related with bile acid and bile salt metabolism, biological oxidation and xenobiotics, of course, ethanol can be considered a xenobiotic. In NAFLD genes, the enrichment biological power are related with endothelin, signal system, and some related with immunological process and metabolism of lipid and lipoprotein. But I cannot read this, and you probably don't. But what about the genes that are specific for each of the diseases? So in red, the genes in small spheres related to NAFLD, and in blue, the gene related with alcoholic fatty liver disease. And in this approach, you can see that even with this difference, all of the genes belong to common pathway, biological pathway. Some are enriched more in alcoholic, but others are enriched more in NAFLD, the specific gene, but all of them belong to the same pathway. So the conclusion of this analysis are that although there may be a bias in the literature, that are very similar in many aspects, except of course for the toxic effect of the ethanol and its derivative ethanol. And this evidence may justify the definition of metabolic dysfunction associated fatty liver disease as a whole. Well, I guess so. But the problem with this definition is we have the patients in this kind of zone, and fatty liver in the end is another component of metabolic syndrome. Then there will be a problem. We try to treat specifically fatty liver disease without taking into account the contribution of the other diseases. The best example is the use of GLP-1 receptor agonist, which the main effect on fatty liver is probably decrease in body mass index. So, well, let's go to the next topic, that is the interaction of OMICS data regarding the pathogenesis of NAFLD. Probably Sylvia have shown you last year many data of this slide. And this is the contribution of OMICS data, the different OMICS data in this knowledge. Genetic variation, of course, is leading by the effect of PNPL3 and another few genes, which can be combined in a score which increase the predicted power a little bit against the traditional risk factors. It's important here to take into account that also mitochondrial DNA variation in gene of the oxidative phosphorylation pathway predict disease severity. Also, there are a lot of knowledge about the contribution of non-coding RNA, which can be divided in long non-coding RNA and short long-coding RNA. Short are mainly composed by microRNAs, miRNA. And of these, MIR 122 and 192 are the most reproducible findings, predicting nicely the fatty liver infiltration, the fat infiltration, even predict NASH and fibrosis, a little bit less power. This can be combined with another traditional risk factor like CK18 with a good area under the rock, which is 0.83. An emerging field is proteomics. There are a little bit less evidence about this, and of course the evidence reflects some expected findings, like the increase of type 4 collagen and procollagen 3, which are obviously the main component of the scar. From metabolomics, there are good information about the predicted power of some common metabolites like glutamine and glutamate, especially the ratio, the contribution of S-adenosylmethionine and beta-in, reflecting the importance of the methyl donor metabolism. Also the contribution of some pro-inflammatory substance, and finally changes in amino acid, including glutamic, of course, and branched chain amino acids. This can be integrated in increase the diagnostic predictive testing power and can induce some intervention response. This is an integration in a network of this knowledge, showing the interaction, of course, of genes, reaction, proteins, and metabolites. This is just a piece of the whole information, showing some relevant aspects like the metabolism of glutathione, transferase, and some related with some kinase. Little bit more focus on the process of urea cycle glutamate and branch chain amino acids. Show this picture, and you see here the importance of liver enzymes in the metabolism. So our view is that liver enzyme is not just a reflection of the organ injury, but it's more an adaptation of the metabolism in response to increase of caloric intake or another oxide such as ethanol. We can analyze jointly the information of genomics and metabolomics, integrating the information of these two disciplines and obtain an enrichment of some biological process. I will show you just the first two, solute carriers mediated transmembrane transport and transmembrane transport of a small molecule. Of course, there are a lot of more biological process, and you see the p-value, adjusted p-value is really significant. Regarding these two first pathway, you see the genes that compose this pathway are some well-known genes like glucokinase regulator, fibroblast growth factor 21, calmodulin, and so on. And the metabolites are some bile acid, some amino acids, and some fatty acids. The second pathway related to transmembrane transport of small molecules the picture is similar, but the difference is here there are some ATP cassettes transported as expected. So what about the role of non-coding RNAs? I will not discuss the contribution of microRNA because this was just mentioned and probably discussed by Sylvia last year. We take in this case a three-step approach. First, we apply system biology with data mining to look at the relation between low non-coding RNA and NAFLD. From this approach, we pick up around 500 genes that was explored against the interaction with the low non-coding RNA. This analysis yield 10 low non-coding RNA. We pick up MALAT1 because it's one of the best known low non-coding RNA related mainly with cancer. Then we see the tissue-specific and co-expression pattern of MALAT. With this previous information, we pass to the wet experiment in 3 stages. In the stage 1, we explored the expression of MALAT in a sample, liver sample from NAFLD in morbid obese patient and against control subject. We replicate the finding in another sample, independent sample from NAFLD related with metabolic syndrome. And then we see the commonality with another chronic liver disease, specifically therapy-naive chronic hepatitis C and autoimmune liver disease. Finally, we try to correlate the expression of MALAT with some of its main target, in this case, VEGF alpha. And then explore in which kind of cells probably MALAT is expressed in the liver. I had problem to passing the slides. I don't know why. Okay. Here you see the expression of MALAT in simple steatosis, again, NASH patient and hepatitis C and autoimmune hepatitis. And you see the significant increase in NASH, again, simple steatosis. This increase is similar, which we observe in autoimmune hepatitis. And in hepatitis C, it's a little bit less, but also significant against simple steatosis. The increase in MALAT is related with lower inflammation score, is related with ballooning score, and also with fibrosis. And interesting enough, the biological process which MALAT is associated to are JAK, STAT, cascade, and response to interferon gamma, and regulation of transcription factor polymerase RNA2. The enrichment show that the opposite regarding the metabolic process, biosynthesis process, and development process, which are underrepresented in MALAT-regulated genes. The correlation of MALAT with VEGF alpha is significant, and the expression of MALAT probably is found in most of the immune cells, mainly neutrophils, lymphocytes, and monocytes. Well, what the information that system bioinformatics can provide us about pleiotropy, I mean the effect of genes in very different diseases or component of the metabolic syndrome. And the picture is as expected. There are a lot of common genes shared by obesity, type 2 diabetes, NAFLD, and hypertension. In this case, we pick up 140 genes that can be also combined with other common processes in this in the metabolic syndrome, inflammation, fibrosis, liver fibrosis specifically, and also this lipidemia. The combination of all of these processes render 50 genes, which are, as expected, very known genes, like PPR gamma, adipokine, and so on. Interesting, these 50 genes can be related with many pathways. This refers to pleiotropy. Cardiovascular disease, immune-mediated diseases, metabolic diseases, even diseases of the central nervous system, or obstetric neonatal disorders like eclampsia. Of course, tumors of different kinds, hematological cancers, and kidney diseases. Surprisingly, also are related with some response to infection diseases. So to try to finalize the talk, I will just touch a little bit the aspect of therapy of fatty liver disease from the point of view of system bioinformatics. I guess you will have a specific seminar about that. This is important because the effective treatment of NASH are really scarce. I guess in 3 years ago there were 2 or 3 clinical trials in phase 4, few in phase 3. So with this kind of approach of integrating this information, we pick up 240 genes related with the same, of course, mentioned pathways, response to hypoxia, immune response, and so on. You can integrate the knowledge of these genes against common drugs and see that these genes are related with drugs really common in the treatment of known diseases, mostly hypertension with the kind of antagonist of angiotensin II receptor type 1. Drug related with inflammation and statins. Interesting enough, we have here supplemental dietary factors like flavonoids and genistein. So the use of these polyphenols probably have a role has a role in the treatment of the disease. Also, we can analyze the putative drug that are trying to be used in this specific trials and see which is the rationale. For example, we can intersect these genes with the genes targeted by elafibranor and see there are 10 very important genes. The same for pioglitazones and vitamin E, selonsertib, etc. So to summarize, we think that integrating OMICS big data can provide an improvement in the clinical information, and this will lead to a personalized medicine. The sum of OMIC big data and clinical feature probably will produce an effective biomarkers to predict the disease and its severity, and this will lead to improvement in patient diagnosis and treatment, improvement in patient stratification and prognosis, specifically rendering more effective clinical trials and discovery of novel pharmacologic agents. Also, repurposing drugs here may be an important next step. Well, thank you for your attention. In this particular project, we just have a few collaborators. The main one is Silvia Sokoyan, that you really know well. So thank Silvia and all of you.
Pablo, your mic is muted.
Yes, sorry for that. Thank you, Professor Pirola. Now we can enter to the discussion of the webinar. If somebody have a question, please, it's the time to do that.
Well, yes, I have a question and a comment. I mean, first of all, I enjoy your presentation. I think that really brings up something new and growing, which is the issue of personalized medicine. The question I have for you, I like the idea to compare NAFLD and AFLD, I mean, non-alcoholic and alcoholic fatty liver disease. The common aspect is fat. And you show quite nicely that the two entities are rather overlapping in terms of gene expression and pathways. The question I have is, what is making common the two entities? I believe that what is in common is the fat, because non-fatty acid, non-NAFLD belongs to the sort of metabolic disease, and AFLD means that you drink too much for your liver. And then the question is, how fat is important in the overall picture? In other terms, maybe fat an over effect, something not related to the disease, but something as the consequence of a metabolic impairment due to alcohol and or metabolic disarrangement.
Yeah, I know you. This aspect interests you, and you are part of the panel of experts that define the new entity, metabolic dysfunction associated fatty liver disease. The answer for your question are really complicated. There are many aspects. First of all, I think the ethanol can be considered from two aspects. First, of course, it's a kind of toxic, xenobiotic substance. Are you hear me?
Yes.
Okay. And this differentiate a little bit the etiology of the disease, or the fat infiltration. But at the same time, it's also an increase in the caloric
Exactly.
supply to the organ, and the organ manage that like other additional caloric overload. So in this aspect, they are the same. This is the first problem. The second problem is we show recently, we're trying to show recently that the fat infiltration doesn't have to be directly cause of the progression of the disease. In other words, for example, the effect of genes on liver fibrosis is not necessarily mediated by fat infiltration. So there are a kind of differentiation in the two pathways. I mean, the concept of the two hits, you know, the first hit is fat infiltration, and the second hit is another hit, blah blah blah. I'm not sure about that. I guess that some individuals manage caloric intake, excess in caloric intake, depositing fat in the liver, but not necessarily progress to the next step, NASH, and so on. There are other factors. In this point, I'm not sure about that NAFLD and alcoholic fatty liver disease are really completely different. I mean, of course, there are many similarities. I try to give you a picture from you know, the concepts of the superposition of the different components of metabolic syndrome. These are real data from the NANES cycle 2015 to 2018. The problem with this database is rare because the alcohol consumption, I'm not sure this was relevated in the good way, because the percentage of people drinking are really low. But from this scenario, again, the distinction between the two diseases is minor.
Thank you. I agree. I mean, I agree that alcohol is a caloric source, and I think that the distinction may be difficult. And
yeah,
I agree with you that fat is not the key issue, but most probably inflammation or redox alteration are the other factors. We don't have to forget that there are the so-called lean steatosis, where people with no overweight, they have steatosis, the other way around. People with overweight, they don't have steatosis. So it's a really complicated story, fascinating. And that's the reason why I enjoy your presentation.
Yeah. But finally, I think that the people that are more exposed to this disease are kind of those that have a lipodystrophy. I mean, people that cannot deposit fat in the adipose tissue. Yeah. So the lean people are, in fact, higher risk to suffer this kind of complication, not only in the liver, I mean, in heart and other organs also.
Right. Yeah, agree. Thank you.
Thank you.
Somebody have other questions? If no, I will do one. Yeah, talking about complexity, practically, if you mentioned that it's more, but do you think that it's more lipodystrophy, the cause or that induce the disease in the liver? I think so that so we will need to integrate more data, not only those regarding to liver. If not, we will need to look also the other tissues, no, in this disease, the adipose tissue and also the subcutaneous or I don't know, pancreas and so we will need to integrate signals from a lot of tissues. This is difficult, but I think so that yes, we need to go to this type of integration, no? Did you agree with this? Do you think that this is yes, in some way we will find markers or the methods to individual novel biomarkers and therapies for NAFLD?
Yeah, I absolutely agree with you. I guess the scientific community and physicians have a biased view of the patient according to the discipline they adhere to. You know, so for a pathologist, the main problem is the liver. For cardiologists, the main problem is the heart. And well, this is a kind of biased view, and I guess the problem is the whole organism, and the study should take into account this complexity. Finally, I guess the main recommendation is don't eat.
Exactly.
Don't eat.
Yeah.
Put a lot.
Follow your diet.
Refrigerator and don't eat. This is
a very sad life. This is a very sad life.
Yes, it is.
I mean, the main way to extend life is restricting 30-40% caloric intake, and this is the well proven in another organism, from the rodents to Xenoharbitum elegans and so on. So don't eat. The problem is difficult. I know we are angry and also enjoy the gelato and so on.
Too many things. Okay.
Yeah.
But, okay, thank you. Somebody have other questions?
I have a question.
Okay.
Thank you very much, Professor. NASH and alcoholic liver disease have the same motivation is the inflammation and fibrosis. Is there a difference in molecular cytokines in the role the inflammation in these diseases? Alcoholic and NASH, the motivation of the cytokines for white cells in an aspect of the inflammation. Is a difference or the same molecular pathway in inflammation for both diseases?
I think, to be honest, I have to recognize that we don't analyze specifically this kind of a small difference. The whole picture indicates that there are common pathways between the two. But specifically, if there is one interleukin in one case and another interloop in another case, I can give you the answer because I don't know.
Thank you.
Okay, other questions? So no. Okay, so thank you, Professor Pirola. And have a nice day. All of you have a nice day.
Okay. You too. Thank you for the invitation.
Bye. Thank you.
Thank you.
Stay healthy.
Yeah, also
Thank you. Bye-bye.
Thank you. Bye-bye.
Bye.